Okay, one of the things that you need to be able to talk about for meiosis is how meiosis actually promotes genetic variation um, in an organism, or actually in a species for that matter. Because asexual reproduction is the offsprings are essentially clones of the parent. But sexual reproduction allows for a lot more different combinations of alleles, and we know that offspring are not genetic clones of their parents. Now, that's important if we're thinking about evolution, because if everybody's the same, if there's some sort of selective pressure that would, affect one individual, like maybe kill them, that could wipe out the whole population. But if there's a lot of variation, genetic variation in the population, there might be a few individuals that are able to um, withstand that genetic or that evolutionary pressure, that selective pressure. And if with that, it means the species in general can survive large selective pressure events better than ones that are clones of each other. So, how does meiosis promote this genetic variation? Well, the first thing we know that in prophase one of meiosis, crossing over, because we get two copies of each, chromosome, right? But each of those copies could have a different set of alleles. Now, the offspring might or might not inherit, you know, all of your paternal or all of your maternal chromosome in the end. So this is, all the different ways. So it could be an entire paternal or an entire maternal chromosome, but they also, because of crossing over, it can kind of mix and match pieces of those chromosomes. So these chromosomes are not the ones that you put into your gametes are not identical to each other. So there's a lot of variation just in the individual chromosomes because of crossing over. And I think we tend to see and for each chromosome, about 3 crossing over events in each chromosome, between the chromatids in a chromosome during meiosis. So that is important. But the other thing is, just even if we weren't looking at crossing over, the genetic combinations since we have 23 chromosomes of all the different paternal or maternal possibilities that you might end up getting, is actually 2 to the 23rd. So that's a lot of zeros if you put it in your calculator. And because the bivalent, or the homologous chromosomes, they line up randomly along the metaphase plate. So if you look in metaphase 1 here, the way that they're lining up, this one has maternal, paternal, paternal, where this one they're mix and match. So there's maternal, paternal, maternal, paternal. So they could go in opposite directions. And just that simple, if you're looking at 2 chromosomes, that can result in a lot of different combinations, right, of possible gametes. Well, then take that out to 23 chromosomes. It could be all maternal and all paternal in the way it lines up, or it could be flipped in either way. So you take that along with crossing over, and it's, an endless amount of variety of each individual egg or sperm cell in that final gamete. So the gametes themselves are super duper unique. And then the other thing is that with sexual reproduction, because we need a sperm to meet an egg, that's 2 unique gametes. What are the chances of this one unique gamete reaching this other unique gamete of this sperm and this egg in all those possibilities? So you really are kind of like 1 in a gazillion. So the 3 main things, if you are asked, how does meiosis and fertilization promote genetic variation? Crossing over creates genetic variation in the chromosomes themselves, like in each of the possible combinations of chromatids in the end. Then, how those chromosomes sort themselves into the individual gametes, that creates, this is called independent assortment, right? That is another layer, right? So first we've got crossing over for the individual chromosomes, and then how those chromosomes sort themselves into the gametes is another layer. And then finally, fertilization as that extra layer on top. So there's 3 kind of main things that you want to talk about. Now, the next thing, we've already looked at karyotypes, but I just want to talk to you a little bit about how we can start to get karyotypes where you have more or too few chromosomes. So either monosomies or trisomies, because there's something that can happen called nondisjunction. So take a look. Here's normal meiotic division. So you've got, and this just for 2 chromosomes, right? So here's, the normal beforehand, we've got meiosis 1, splitting of the homologous chromosomes, then splitting of the sister chromatids, and each of the gametes have the same number of chromosomes as the parent. Well, you can get nondisjunction that happens in during anaphase of meiosis 1. So in this case, two of the homologous pair doesn't separate from each other. Normally, this one would go this way and this one would go this way, but these two separate out in that direction. And then you can see what ends up happening is that you get an additional chromosome in two of the gametes and too few chromosomes in two of the other gametes. So if this one gets fertilized, you could end up with a trisomy, right? When the sperm meets the egg, then you would end up getting 3. That nondisjunction can also happen during meiosis 2. In this case, it's only going to create one trisomy and one monosomy possibility, and then the other one divides normally. So if all goes well, you would have the normal division. So you've got the haploid number. So when the sperm meets the egg, then you'll have the diploid number, in this case of 4. But here, if a normal sperm, say a haploid of 2, meets this haploid of +1, then you'd end up getting 5 chromosomes instead of 4 for the individual. So just realize that you can get this separation of chromosomes that doesn't happen the way it's supposed to. That's called nondisjunction. So if you want too few or too many in a particular gamete. And the older that you get, that increases the likelihood of nondisjunction occurring in gamete production. That's important because when you're thinking about having children, the older you get, the higher probability you would have a child that has, say, trisomy 21, like Down syndrome. In fact, it's pretty significant, the differences as you start to age. So there is a lot of different types of tests that you can take if you're pregnant to see if the fetus has, the growing zygote or fetus has a trisomy or a monosomy. It used to be very common that they would take amniotic fluid. So here would be the growing fetus, right? And there's this fluid that it's swimming around in, right? And this fluid is going to have lots of cells of the baby. They stick a giant needle in through the mother's belly and withdraw some of this amniotic fluid. Then they can coax it to go into mitosis, and they can make one of these karyotypes, right? These karyotypes that we see. So they use amniotic fluid to make a karyotype. The other way that they can get those cells out, they can do it even earlier. This happens generally around 15 weeks, but you can do it, I think this is early as maybe 10 or 11 weeks, where you go in and you take some of the chorionic villi. We're going to look a little bit more closely when we're doing reproduction about what that is, but it's these little finger-like projections that the fetus fetal tissue puts into, interacts with the mother's tissues. And so you can take some of those, some of that chorionic villi tissue, and which is going to have the fetal cells in it, and you can make a karyotype from that as well. So when you get the karyotype, then you can see how many chromosomes there are. And generally, as a woman gets older, they tend to more want to have these done just to make sure that they can prepare to know if their offspring is going to have the right number of chromosomes or if there's any chromosomal abnormalities. But there is a downside to it. Besides the fact of having a giant needle stuck through your belly or a giant suction tube inserted through your cervix, is that it also runs a risk of miscarriage. So there's a risk of miscarriage of 1% for amniotic fluid withdrawal, and for the chorionic villus sampling, it's a risk of miscarriage of 2%. However, technology is getting even better and better, and they can now, so take a deep breath, ladies who are planning on getting pregnant, you can now take a sample of your blood and actually pull the fetal DNA out of your blood at about 10 weeks and make a karyotype that's pretty darn accurate. Now, it's not 100% diagnostic like these two would be, but it doesn't put the fetus at risk, and it's actually quite, it's pretty darn accurate. If they get an inconclusive result, then what you can do is have one of these done to get a final, yes, for sure, that's what it is. So new technologies, they can actually piece together fetal DNA out of your blood to make a karyotype instead of doing these procedures. But these procedures would still get used to confirm if the original one with the blood samples came back as being a high risk for that. Okay, so you need to be able to talk about how we can determine if non-disjunction has occurred through being able to analyze a karyotype and also talk about how we do that. So through amniotic fluid withdrawal and through chorionic villus sampling, and then kind of talk about the fact that there are some side effects and risks to that.